from the College by the Lake, Coeur d'Alene, Idaho. Local, regional, national, and international guests discussing the issues and topics affecting the way you live are on Forum, the North Idaho College Public Forum, with your host and moderator, political scientist Tony Stewart.
Welcome to the Public Forum. Our subject today is a very, very important one, and we're most fortunate to have two very professional individuals to discuss this subject in a most articulate way. We're going to discuss the issue of viruses, and it's always certainly been an important medical question, and there's a lot of attention given to it, and our guests can talk about this in a very clear way. And we're going to divide the program into an overview of viruses, and then we're going to look at some of the broad spectrum of the issue of viruses, and then the latter part of the program talk about treatment. I am so pleased to welcome two guests today, and they were very gracious with their time to come in from a busy schedule to do our program. First of all, to my far left is Dr. Frank Kimball, a medical doctor, a pathologist. He holds his medical degree from Johns Hopkins University. Dr. Kimball, welcome to the program. Thank you. And I'm equally happy to have here today Dr. Bernie Bernard, who is also a medical doctor, a pathologist. He holds his medical degree from the University of Oklahoma. He also has a PhD in the field of microbiology that he received from Kansas State University. Dr. Bernard, welcome to the program.
Thank you.
I also would like to point out that both of our physicians today are members of Pathology Associates of Spokane. Dr. Bernard, I will start with you on the very first question. And as I introduced the issue of overview of viruses, would you take a minute for we laypersons to define what we mean by viruses, and as you do that, clear up for us the confusion, I guess one would say, or contrast with bacteria or microorganisms?
Well, I'll briefly mention a couple of things, and then Frank will maybe talk about some other issues. Viruses are the smallest of the traditional microorganisms that we deal with in biology and medicine. Compared to bacteria, they're smaller by a factor of 1,000 in some cases, although some viruses can be maybe only half as small as a bacteria. So they vary quite a bit, but in general, they're small. And the difference between bacteria and viruses from the biological point of view is the ability of viruses to go through filters that could withhold bacteria. And historically, we knew that there were agents smaller than bacteria that caused disease, and then it was maybe in the 1930s and 40s that we began to realize that it was really viruses that was that unknown agent. And now we find out that as of today, we're probably looking at somewhere around 800 to 820 different types or subtypes of viruses that infect humans alone.
And so our modern technologies had a lot to do with even discovering those. It took that to identify them, did it not?
Right.
Dr. Kimball, as Dr. Bernard has indicated, there are a lot of different kinds of, a lot of numbers of viruses. Are there others that we probably don't know about, or how do we explain the fact that we hear new terms, or it seems like the cropping up of viruses? I suppose that might be true in bacteria too, or is it?
Well, I think that the world of viruses is a world we're just beginning to get into, and I think it's interesting to think about viruses in a Darwinian sense as well. They represent, as Bernie said, the very smallest particle of life, and they may not even be life, depending on how you define it, because the only component of life that they have is the ability to reproduce. They have no cellular machinery to make proteins, growth substances. They can't generate energy. They're obligate parasites to cellular organisms. And in fact, certain viruses, if you put them in the right environment, form crystalline structures, an attribute that we usually associate with inorganic materials. So they're certainly at the cutting edge of the interface between life and inanimate existence. And I think that we've only been able to envision them since we've had an electron microscope capabilities. And then we now have a variety of techniques that let us either imply or actually physically demonstrate their presence, and we can measure them and look at them and so forth. But this is the newest area of direct observation, as Bernie said, since probably the late 1930s, early 1940s, and electron microscopes really weren't available with any degree of efficiency until, I believe, in the 1950s. So this is just a new, and like everything in life, it's on and on to greater and greater levels of complexity. I think we're just beginning to really classify and have an understanding for viruses.
Along that line, Dr. Bernard, the fact that we've not been able to identify them or know as much about them because of lack of technology, have they been around and operated as actively as they are now, like bacteria is done, and we just know more about it now, or is there an increase in the activity and development of viruses?
Well, I think there's some evidence. I think that viruses have been around probably as long as humans have been. I think there's a constant evolution of microorganisms, just as there is probably in man and every other form of life. So I think there's gradual changes that occur and have occurred through time. Viruses commonly begin to exert their greatest force when the host or the body that becomes the host of the virus, which is really a parasite, it has to have a relationship, a parasitic relationship with the host animal. When those animals get stressed, and it's a matter of susceptibility and it's a matter of transmission, transmittability. And so it's not uncommon that we experience common colds more often in the fall and winter than we do at any other time of year. That's not an accident. Those cold viruses are probably around all the time, but the fact that we begin to house ourselves in smaller and smaller areas in higher and higher concentrations in the winter because of weather, we tend to have bursts and valleys about the common cold activity. If you look at common cold in the equatorial countries, there is no seasonal variation. They get a colds interspersed in a fairly constant fashion across the year, while in a temperate area like the United States, we experience some of these fluctuations in incidence of disease. And that's a matter of availability of susceptible hosts.
So the environment can affect what is happening.
Right. And I think as population increases in the world, that begins to create a fertile ground for the emergence of new disease. And one of the things that has been in a lot of people's mind over the last 2 to 3 years really have been this phenomenon of emerging infectious diseases. And the question is, are those new diseases or old diseases? Mm-hmm. Well, I think most the data suggests that those diseases have been around. Those agents, whether they're bacterial or viral or fungal, have been around for probably a long time. It's that something has happened in our world. Many times it's population density or nutrition or things like that that create the environmental situation that allows those viruses to take hold, flourish in the population, and cause disease. And then if they cause disease in a focused enough fashion that people all have the same sorts of symptoms, or many people in a certain area get a disease, then the epidemiologists and the biologists get busy and start investigating those diseases, those situations, and begin to find new emerging conditions.
So there are more hosts as the population goes up.
It's interesting to think about this problem in terms of what the virus is trying to do. And viruses, like all living organisms, are just seeking their niche where they can survive well. And viruses are very varied, and Bernie will talk about that later, but they have a variety of ways in which they can maintain themselves. They can just invade cells, reproduce in massive numbers, destroy the cell, and hope that the released baby viruses will be picked up by another cell or another organism. they can invade cells and actually incorporate themselves into the genetic material of the cell and be latent. And that's the sort of thing that we worry about in part of the problem of where do viruses go in between epidemics?
Mm-hmm.
Do they? And we know that many viruses, like the AIDS virus, hides in lymphocytes, and certain tumor-causing viruses hide in the cell, and they actually make the cells immortal. They're tricky little devils. Mm-hmm. And another approach is to just utilize the cell transiently and then emerge from it without destroying the cell. And these are all different modifications of the niche. And I think it's also important when we're worrying about, or there's sort of a fear-mongering of killer viruses, Ebola viruses, Marburg viruses, that any parasite that kills its host is a failure in a long-term biologic sense. They don't really care how bad they hurt the host, but they like to have the host be either survive and continue to provide the environment in which they can exist, or if they're going to kill the host, then they have to darn well have some other host nearby. So it all gets into an interplay of proximity and survival and a variety of other factors.
That's a good bridge for us to the other area we want to deal with, was what we call the broad spectrum of viruses. We, at the beginning, identified the hundreds of viruses. Dr. Bernard, could you take us through what we mean by broad spectrum viruses with some examples? I know we want to talk about Hantavirus and the AIDS or HIV, and we've heard of Lyme disease. So I have a two-part question. One, walk us through that, and then we'll talk a little bit about where they come from. Do they come from other animals to humans or from human to human?
Okay, well, as I mentioned, there's something like 800 known types or subtypes of viruses, and viruses cause disease by first attaching to and then invading into cells. So the type of disease that a virus produces is almost entirely dependent initially on what cells have receptors on their surface that viruses can attach to. And maybe I can use it as an example. A polio virus that fortunately we don't have much disease with anymore, and actually maybe moving toward disappearing, entered the digestive tract and attached to the lining cells of the intestine. It tended not to involve the respiratory tract, and the feeling is that the receptors on the cell surface able to allow the virus to attach did not exist in the respiratory tract, but did exist in the gastrointestinal tract, in the intestines. So once the virus was able to attach to the intestine, it then infected this new host. So if it entered someone's body by existing in the water that they drank, or was on food that they drank or ate, then the virus would have the opportunity, if it could survive passing through the stomach, to attach to the intestine, invade those cells, cause damage, sometimes causing clinical diarrhea, so that the patient knew that they were ill from some problem. But then once the virus got into the tissue, it would continue to flourish, would spread then in the bloodstream throughout the body. And we know that then the viruses went to the central nervous system and caused its problem that led then to the paralysis and the more serious problems. So that's a situation in polio virus. We can look at influenza. That's a different virus. It's a respiratory virus. Its focus of initial infection is in the upper airways. And so if one inhales that virus on droplets from someone coughing or sneezing, if one has it on, or gets it on your hand from contacting either an object or another person with a virus, we can eventually get it into our respiratory tract where the virus can attach to the tissue cells lining the respiratory system. Once there, it can invade those cells. It can then proliferate, invade the bloodstream, and be distributed. For practical purposes, influenza disease is pretty much limited to the airways. I mean, pneumonia is one of the main problems that influenza produces on the severe end of disease. Lesser disease of laryngotracheal bronchitis or upper airway disease certainly exists. So all of the fever, the chills, the muscular aches and pains of influenza virus all follow after the virus gets its foothold, in its case, in the respiratory system.
So really what we're saying here, this is fascinating to me, and I thought of it in that way before. It depends on how it travels, how contagious it is, or how many people may get it.
Exactly.
There's another aspect of this too, and what we're really describing is a war, an ongoing, constant war between forces external to ourselves and our own defense systems. And what we've left out of this equation is that we've talked about how the virus attacks and establishes and reproduces and spreads, but you have to remember that the virus is up against a very potent defense. And the first thing it has to do is get through the mucus substances that line all of our, either we have tough skin or we have mucus on our inner linings, and those mucus substances prevent attachment to cells. So they have to develop systems in their virus apparatus or armamentarium that will somehow break down the mucus or the substances that inactivate them, allow them to get to the cell. Then there are antibodies at two levels. There's antibodies right on the front line, right in the mucus or on the surface, that are called local, and those, dependent on the amount of exposure, are able to key in or not key in on the attacking virus and inactivate it again. So it's got to get past the antibodies locally, it's got to get past the toxic substances the host produces, and then if it makes it into the cell, the next round is to reproduce, break out of the cell, get into the bloodstream, spread to other susceptible sites, except in the blood is another whole level of antibodies which are geared up, if they've been exposed before, to eliminate those viruses. So it's not real easy for the virus, and that was witnessed to the fact that most of us walk around in good health most of the time.
And therefore, the better your immune system is, the more likely
Absolutely.
the virus won't succeed.
And therefore, the greater stresses that you have, theoretically, the less effective your immune system is and the more susceptible you may become.
So you can do an awful lot of things. You know, you can lower your stress, or you can not be in the house all the time with the influenza types. Something else that you really made clear, and that was like with influenza and so forth, it's in the air and someone sneeze and breathe it in, whereas you have other things you've talked about where it's some water, food that you take in, if we're more aware of that. I'm thinking of the Hantavirus now, and I understand that's from rats and so forth. So if you're aware of those things and can eliminate that in your environment, or at least in your home and so forth, you have really attacked the problem, have you not?
Correct. You know, when we talk about the variety of viruses, we sort of touched on in a couple of aspects, but there's a whole series of sort of issues, as you mentioned, the manner in which we contact a virus, or the mode of transmission. And as you mentioned, Hantavirus is associated with rodents. And since the Korean War, we became aware of the existence of hantaviruses, although it was some years after the war that the virus was actually characterized and identified as a separate and unique agent. And now there are somewhere in the range of 7 to 8 different hantavirus subtypes that we know of. All of them are rodent transmitted. Some are transmitted by rats, some are transmitted by voles, which is another rodent. And in our particular case, the Hantavirus that recently appeared in the Southwest and have had now sporadic cases in, I think, 21 states, including Idaho, is spread by the deer mouse, which is a common rodent in our part of the world. In the case of the deer mouse, populations flourished in the Four Corners area in the Southwest because there happened to be a heavy rainfall year, and the pinon nuts from the pinon trees were very high in number. The mice feed on the pinon nuts. Their populations increased. They began to find new living niches, which included homes and houses that the people lived in, and therefore the opportunity for contact between excretory products of the mouse and the human developed. And then when areas where the mice had urinated or left fecal droppings were disturbed and aerosols produced, the humans then had the opportunity to breathe that in, and the infection could get its foothold.
Dr. Kimball, in relation to what Dr. Bernard is saying, that I'm thinking then of another problem, and that's HIV and leading with the immune system being attacked to AIDS. But again, we're talking about a virus here that's much more difficult to find a host. That is, you can't get that as easy as you can, obviously, influenza. So would you speak to that issue? And another part of that is, it must be obvious that some viruses are attack more deadly, the immune system cannot resist, than others. In other words, in a layman's terms, I would say, are some viruses much more vicious than others?
Oh, I think it's true that some viruses are more vicious in the sense that they make us sicker or kill us. The way in which they do that is probably not intended to be vicious. With the AIDS virus, and falls into a chronic latent virus that homes on a certain kind of lymphocyte. It affects its damage in a sort of indirect way. It has a propensity for what we call helper T lymphocytes, and it is able to get into those lymphocytes once it's entered the host through an appropriate break in that defense system, and become a part, really, of the genetics or the DNA code. Well, is it a DNA?
It's in the RNA virus.
RNA virus, but
a copy of DNA is made from it.
Anyway, through the course of time, it lessens and eventually obliterates this population of essential helper lymphocytes, and thus paralyzes the host's immune system. And then the host doesn't succumb to the virus; they succumb to the secondary infections that, if they had a normal, healthy immune system, would not be a problem. So it's sort of an indirect relationship. But again, that's a niche that particular virus has found. Now, we say we have an epidemic of AIDS, but it's not an epidemic in the sense of influenza or a common cold or that sort of thing, because the mode of spread is so much more difficult, and we wouldn't have begun to have an epidemic until good quality needles were developed. Disposable, high quality, easily available needles. Well, that's a technological change in the environment that allowed this particular virus to spread more rapidly. With most viruses utilize, if they're going to be of epidemic quality, they're going to utilize either air or oral ingestion, and air is the most likely, and the most common epidemics and pandemics are related to aerosol spread, because human beings breathe in and out air about 20 times a minute, and so you have a pretty good sampling process and exchange process going on. So I don't know if that answers your question, yes, but it does point up the diversity of the way in which viruses can operate, and yet the end result to us is we get sick, or we get deathly sick, or we die, and we don't really appreciate that there's a whole variety of different processes going on.
One of the things we promised our viewers too, and we're on the edge of that right now, and it has to do with treatment, and I think you've done such an outstanding job of talking about the mode that's used for the different viruses, and also how they can attack cells or different parts of the body. You always are faced with we who come to you and we're now sick, and you have to try to treat the viruses. I guess I have a very general question, and that the viewers should certainly be aware of, and that is, on some of those viruses, your treatment is fairly immediate and very effective, and others, it's very difficult. Why are different viruses so different in treatment?
Well, the viruses in general have fewer metabolic sites for action. Frank mentioned differences between the bacteria and viruses, and bacteria have their own kinds of intracellular organelles to do their metabolic thing, to grow and reproduce. And bacteria on the genetic tree are quite distant from humans, so many of those subcellular organelles are unique to bacteria and are not shared by humans. Those differences allow targets to be developed that can affect the bacteria and, relatively speaking, not affect the human being. And antibiotics do that. Bacteria have cell walls. Human cells don't have cell walls. Penicillin was a antibiotic that was discovered that acted on the cell wall production and prevented cell walls from being able to develop. And that's why penicillin worked on bacteria. Since humans didn't have cell walls, penicillin is relatively non-toxic. Other antibiotics act at many other sites. The problem with viruses, as Frank mentions, they don't have their own intracellular machinery. They parasitize the human cell and divert the human cell subcellular organelles to do the virus bidding rather than the normal metabolic activity of our own cell. So the targets are not unique. They're our own. So that if one develops a drug that's going to affect a virus, the chances are there's going to be some toxicity to the human body because the virus is using the human cell organelles to do its growth and reproductive activities. So we don't have this genetic or developmental differences to be able to use as targets. So that makes it much more difficult. There are some antibiotic or some antiviral agents available. Some work very effectively, and I can think of respiratory syncytial virus, which is a fairly common respiratory virus in children that can cause severe airway disease leading to airway obstruction, and if not treated and controlled, can lead to death. There is an antiviral agent available to treat those patients that they respond quickly, and it's not very toxic to the human, so it works well. In the case of influenza, there is a medication that can help block the infection from occurring. It's called amantadine, and it's commonly used in settings to protect people from getting the disease, because it's been shown that someone who already has influenza and you begin to administer the antiviral agent, you don't really change the course of their disease. They are going to progress on through and get well at about the same rate whether or not you provide the antiviral or not.
I've just got a signal we're out of time. I really wish we weren't.
No.
I hope you two gentlemen will come back and let us do another show in the future. You have been extremely informative, and we thank you very, very much. And thank you for your time. Ladies and gentlemen, our guests have been Dr. Frank Kimball and Dr. Bernie Bernard from the Pathology Associates of Spokane, talking about viruses, and I'm sure you have found this program very helpful and informative for you, and we will invite them back in the future. And I would like to invite you to be with us again next week at this same time when we will discuss yet another subject. Until then, please have a good week. I am Tony Stewart.
The North Idaho College Public Forum was videotaped live from the studios of Instructional Technology on the campus of North Idaho College for viewing at this more appropriate time. We invite you to join us again next week for another all-new edition of the North Idaho College Public Forum on this public television station.